Welcome back to The Issue on the ASGCT Podcast Network. I'm your host, Emily Walsh-Martin, and today we have a very special guest, Chris Choi. Chris is the Senior Vice President of Industry Partnerships and Core Facility Director for GMP and Tech Ops at Roswell Park Comprehensive Cancer Center, as well as an Associate Professor of Oncology. Chris and I talk about cell therapy development and manufacturing and how the growing Roswell Park Center is aiming to support sponsors in the CAR T and cell therapy space. We also talk a lot about our personal experiences on cross-functional teams and how the research, manufacturing, and clinical elements of a program must be knit together for success. But first, it's my annual plea for long-term clinical data. Seriously, though, abstract submission is open for the annual meeting in New Orleans in 2025. So while you daydream about beignets and jazz, start penning your abstract. Hope to see you there. But your first step is to visit the website, annualmeeting.asgct.org/abstracts. And now, on to the show. All right. Welcome to The Issue on the ASGCT Podcast Network. Chris Choi. Chris, you're the Senior Vice President of Industry Partnerships and Core Facility Director of GMP Tech Ops and the Associate Professor of Oncology at Roswell Park Comprehensive Cancer Center. We are so happy to have you here today. And I want to talk to you about where you see the field of cell therapy development going, and as well where manufacturing is heading, and also about the role that Roswell Park hopes to play in the future of this development for their patients. But first, can you tell us a little bit about your background?
Thank you, Emily, for having me. It's a pleasure to be here. And thank you for having this podcast. It's a fantastic medium to educate the broader audience on this really new and burgeoning field of cell and gene therapy. Just a little bit about myself. I started in cell and gene therapy. I'm going to age myself back in 2000. My first job out of college was working in an academic GMP facility with Dr. Don Cohn, who was a former president of ASGCT. So I worked on the initial ADA SCID trials, the stem cell gene therapy trials, as well as the hemoglobinopathies, X-linked SCID, et cetera. So having been through that and then to where we're at right now is just really amazing. So I did end up getting my PhD with Don Cohn, and then worked in academia. You're a grad student, you're a researcher, and sometimes when you need to do GMP manufacturing, you do GMP manufacturing. So after my PhD, I followed into UCLA and worked on the cell and gene therapy trials there with the team there. And we had great faculty and mentors, Dr. Tony Ribas, Don, Gay Crooks, et cetera. I did end up going to business school, so I went to MIT Sloan. That's really where I got my operations chops. And I was fortunate enough to be recruited to Roswell Park as to build their first GMP facility. I did leave for a stint, went to Kite Pharma, learned a lot about early as well as late stage and commercial life cycle. Then went to CRISPR Therapeutics, where I had process development, facilities, utilities, systems, engineering. So I had process design, MSAT, as well as Fuse reporting into me. Worked on the IO products as well as Casgevy. And, Roswell, with New York State's involvement through Empire State Development, is expanding the new facility to 20 rooms. And one of my colleagues and really mentor and friend, Rainier Brengens, who recently won the Warren Alpert Prize for his contributions to CAR T therapy, was already here at Roswell. And, he had this opportunity. So I thought it'd be great to work with friends and really build what we're trying to be a destination for cell CAR T therapies.
Absolutely. So that's That's quite a history. And I think for me, it sort of reinforces the fact that there are many paths, right, to joining the field of gene and cell therapy. You found a very early path.
Yeah.
There's a bunch of us who spent, for instance, a decade in small molecules before we, crossed over into this field. But you, having followed that early path, have really seen, the field evolve over time, and you have that vantage point. So we'll definitely double click on that experience. It's also interesting, right? Because you and I both passed through CRISPR therapeutics and worked on Casgevy for a time, but non-overlapping. And I think for me, like, it still amazes me, right? It absolutely takes a village. Casgevy moved very, very quickly from development candidate nomination and into the clinic, and then now being a commercial product. But probably over the course of that 7 to 10 year time period, thousands of people played integral roles in helping shape how that product looked and moving it into the clinic. And it just reminds me that for every product that's worked on out there are these phases of development: early, sort of middle, and late. And, in early times, you maybe don't know if something's going to work in the clinic, and you have to decide how much to invest. In middle times, you maybe start to have a sense of what's working, maybe what's not working. And you're trying to fill in the blanks that were left in the early phase. I'm just curious about your general experience over watching products from early stages to later stages, and how you, I mean, do you enjoy that challenge of inheriting something where you're trying to fill in the blanks and do that next step, or do you like the early phase better? Like, what do you prefer?
Yeah, it's quite different. The goals for early, late, and of course, commercial, when you look at the life cycle of a product. And so when you take this quality by design approach, you start off with your target product profile, then you have your quality target product profile, and these are really your CMC requirements. But throughout the life cycle, and going, as you mentioned, the mid and then the late and commercial, it's really about increasing your product knowledge and your process understanding. And early phase, you don't really know much about the product. You have the research data, you have the mouse data, but then how do you translate that, and how do you take that into human? And so the great thing is the FDA already has this structure, right, with the phases of a clinical trial. You have phase one, that's safety. And what's interesting about cell and gene therapy is phase one, you also look at efficacy. So it's, a lot of these trials are phase one, or phase 1/2A, or phase 1B. There's all these different types of combination. And I think for the earlier cell and gene therapy or cell therapy trials, it really went from a phase two pivotal to an application, to a BLA application for the US. So I think the goals are different. For early, it's really about safety and seeing if something works. And then later on, it's really about consistency and manufacturability. So you did a great job, Emily. It worked.
Well, and what's interesting, right, is that you said something important there, which is manufacturability and sort of consistency, right? And that's something that the biggest challenge with cell therapies in particular, right, is the person to person differences, right? You can control a lot of things on the manufacturing side, but at the end of the day, the person to person differences can have big impacts. And what interests me most about that is sort of at a place like Roswell, where you guys are building out this new facility. You're going to have I think you mentioned to me in our discussions, like 20 rooms. So it's going to be a very large facility. You're going to have a lot of cross patient data that may really help you guys to understand how to minimize the patient-to-patient differences in the hopes of having a consistent product. I mean, is that how you're looking at it with this new facility that you're building out?
Yeah, you hit the nail right on top of the head. And from experience, and as you know, as you're going to commercial, it's really about the operating range. You have your normal operating range, you have your proven acceptable range, and then you have
Hmm.
your edge of failure. So that's your control strategy for late stage. Early stage, it's more about the specs and the limits. And you're absolutely right. From experience, doing characterization for commercial products and products that are in late stage and about to go commercial before the PPQ, the primary source of variability in autologous cell therapy manufacturing, and even allo, it's the donor or the patient apheresis product. So statistically, that's the biggest, that's where you see the most variability is the donor
Right.
or the patient product. And so what Roswell is building with our 20 rooms, and the 20 rooms, we're going to be making viral vectors. We also plan on making plasmids, but we're currently making viral vectors. And our new 14 rooms that we're building for cell manufacturing is going to be done and complete and ready to manufacture as of March of 2025. And so we're partnering. We do already have a couple of biotech partners, and we have our own internal clinical trials. And from all the data that we're gathering, we can iterate working with partners, working with our scientists, physician scientists here at Roswell to really increase that product knowledge and also process understanding. So we can decrease that variability and increase manufacturability.
Yeah, that's the dream, right? So tell me a little more about the Roswell facility and sort of how do you guys imagine sort of partnering with potential partners to help bring their products to reality?
Yeah, thank you for that question. As I mentioned earlier, we want to be a destination for CAR T and cell therapies. The facility was originally built in 2011. We started as a one-room clean room facility, and that's why I was originally recruited to Roswell Park. We had a shoestring budget, and Dr. Marco Davila, really a giant in this field, still teases me today about starting up with a one-room facility. We used to call it GMP in a box. That's yeah. But it really took, and this is before people thought cell therapy worked. So if you went to a conference and you would ask, and this is when SGCT I think was maybe 1,000 people or
Right.
2,000 people at the time, and you ask people, does cell therapy work? You see a couple of hands go up, but now with multiple commercial products, everybody believes it. So we went in 2013, we built a viral vector production facility, 2 room, with an additional 4 room cell production facility. And with recent partnership or investment with New York State, the Empire State Development, they've now contributed $30 million to build 14 new clean rooms to work with partnerships with industry as well as academic partnerships. The way that's structured is we want to be a partner in the truest sense. So if you have an idea, we can do the preclinical research, we can do the tox studies, we can also do the first in human, and we can take you to pivotal. And we were trying to build all the capabilities in-house from analytical development, QC, viral vector manufacturing. And we're also looking at AAV in addition to already we're making lenti as well as gamma retro. And we're going to make a plasmid production facility as well.
Wow. And, I want to double click a little bit. Former listeners to the show will know that I love analytical development. So if you don't mind, let's double click a little bit on that. Just because
Sure.
when we think about clinical trial, like running a clinical trial with one of these products, like that is always on critical path. And it's never comfortable. And but it's always the case, right? You need to have
Yeah.
some level of release testing on a product, especially for a CAR T product before you can dose it. And how do you guys sort of think about that? What are you trying to do to, as much as possible, sort of speed that initial testing of a product that you've manufactured?
Yeah, so we're very fortunate. Dr. Brenjans was co-founder of Juno Therapeutics, so he's been doing this for quite a while. So there's a lot of data and knowledge. And myself, I was at Kite and at CRISPR, and we have several others that have worked in industry, and now we're all here in academia. So because of that, we already have a fairly mature knowledge base. But even before that, taking a quality by design or risk-based approach, we start off with our target product profile. Then you have all the inputs, right? Material, and you have your quality target product profile. So we do use that same structure. So we have a product quality risk assessment. And so we're looking at, okay, what are our quality attributes? What are our release in process controls, et cetera? And so with that, we're taking the historical knowledge of having been through early phase and late stage, and we try to engage the agency as early as possible. So because we have a structure and we've done a lot of CAR T studies as well as other cell therapies, dendritic cells, et cetera. So we have already an established base of assays that we know that the agency wants to see, but every product is different. Every reviewer maybe have different inputs from the agency. So we try to engage them as soon as possible. And with that input, working with the agency, if they want something else for us to measure or look at, or we'll work with the agency and our early stage scientists in developing these assays. So that's assay development. So that's analytical development. Then after that, does it actually work? Does it work the way that you want it to? So that's assay qualification, right?
Right.
So that's another level that you have to work with partners with the research group as well as our analytical development, where it's qualified within our analytical development group. Then at that point, it's okay, can we use this as a test for the product? And that's where we transfer it or tech transfer it to our quality control group. And, for early phase, the agency really expects you to qualify the assay. So you're looking at specificity, accuracy, linear, et cetera, right? Precision.
Right.
Later on, when you're going to late stage, and definitely when you're looking to go through PPQ, you have to validate. And that's where you pretty much do everything.
Right. Exactly. Yeah. Do everything again and, backwards and forwards for sure. So, let's maybe delve a little bit deeper into that, because I think one of the key concepts in my experience, especially when you're talking about oncology drugs, is the sort of the process from vein to vein. So basically from the time that you apheresis a patient and harvest their cells that you're going to use as your beginning raw material for manufacturing to the time that you're able to provide it back, this is like a super critical time to have it be frankly as short as possible, especially when you're dealing with aggressive solid tumors or blood cancers where the patients are going to be progressing, and you very often can be in a situation where they totally qualify for the study and are healthy enough to proceed when you do the apheresis, but then things sort of progress quickly after that. And so how do you guys think about that vein to vein concept and helping the sponsors to really move their products as quickly as possible back into a patient?
Yeah, I think a lot of these companies have 1-day processes or 2-day processes or 3-day processes, where originally it was 7 to 10-day processes. So that's really helpful in terms of vein to vein time. But going back to the QC test and release tests, that's, I think, the longest, the sterility essentially takes.
Right. Yeah. How many days is the sterility again? So
it varies depending on what you're doing. If you're using US clinical methods, but on average, you have bacterial is anywhere from 10 to 14 days. And then if you do culture method, there's obviously rapid methods that you can do as well that are PCR based. But so it really depends on the product, but it could be up to 21 days.
But a 14-day assay takes 14 days, right? Like that's like, there's no compressing that except with some of these rapid orthogonal assays. Like you were
like a back
about.
T versus doing, yeah. Mm-hmm.
So go ahead, tell us more about how you guys think about that and helping sponsors to sort of navigate that.
Yeah, I mean, I think the one of the things that we're able to do here by having everything in-house. So again, you can control manufacturing, and there are 1 day, there are 3 day processes, and I think the data shows that statistically they're just as good, and from a 7 to 10 day process. So you're doing the expansion in vivo inside the patient. So the way that we're thinking about it is we can, like I mentioned earlier, do everything in-house. So rather than sending it out to a contract testing organization, we can do these assays in-house. If there's an assay that we can shorten, for example, using the BACT or BACTEC versus doing a culture, and again, instead of doing a culture, doing a PCR method, we work with the agency to figure out if this is suitable for what we're looking at or the study that we're doing. So again, we're looking at all the different options, and of course, orthogonal assays is another method that you can use. But even before that, I think being able to do everything in-house really shortens that vein to vein time. And we talked about finding a good target. So outside of that, one of the things that we want to do is being able to find, well, what's a good target? Why does it work, or why doesn't it work? So all of these we work in parallel, and because
Mm-hmm.
we have researchers as well as operations type folks and physicians all housed. So I have Rainier and Marco, Dr. Brengens and Dr. Davila. We're all on the same floor. Our offices are right next to each other. So if I have a question or if they have a question, I could just go right over and knock on their doors. So I think having all of these folks, as you mentioned, involved in manufacturing and getting a product commercial all on the same floor, all next to each other, thinking about these sorts of challenges, it's helpful for our clients and for people that want to manufacture within our facility.
Yeah, no, absolutely. And I think you're right. The whole certainly in my experience, there are pros and cons to having the analytical in-house versus out, but the real benefit of having it in-house is if something goes wrong, right, and you need to retest, you can deal with that like more quickly than figuring out at your outside analytical company has a problem and now you've got to ship, and so that's another couple of days to get the new sample in, et cetera, et cetera. It does make you guys in theory more resilient to when stuff happens, right? This is not this isn't it's still at the borderline of art and science, right? Where assays are good, but still stuff's going to happen from time to time, and being able to react to that, that's certainly a benefit. I guess from your perspective, what else are you guys trying to build in as you build out this new facility and these new rooms that's going to really streamline handoffs between departments and knowledge transfer and that sort of thing? How are you guys building out to achieve that?
Yeah, that's a great question. As you can imagine, when you're in academia, you don't have the resources that you would necessarily have at a biotech company. When you're at a Genentech, you have a lot of resources and teams that are specialists in that area that they're working on. So you have one person assigned to stability, for example. But in academia, there's smaller teams. A lot of our funding is from grants or from donors, et cetera. So where do you find that balance of having that speed and expertise in industry? And we have our own expertise in academia. Don't get me wrong. We have brilliant minds. People have great scientists, Warren Alpert Prize Foundation winners, et cetera. But one of the things that we're trying to build is that nice balance between industry and academia. So our director of quality is a former FDA inspector. Our operations folks, we were able to recruit folks from industry, from Amgen, et cetera. And the way that we're structured is we do have a separate, when you look at our operating model, we have a governance structure that might be similar to industry, essentially. In terms of capability, we have, as we mentioned, analytical development, QC, quality assurance. We also have these business operations type capabilities, technology and material, enabling capabilities, people that have, experts in Six Sigma and Lean, as well as business management. And we have a separate supply chain group, process sciences group, MSAT group. And, that you were talking about OSs previously. really that MSAT group really handles a lot of those investigations, process
Right.
monitoring, data analytics. So that's some of the structure that we're building. And really it's the operating model, right? We have CMC teams, we have CMC leads, we have project managers. And so all of those capabilities that I mentioned seems like a lot of people, but a lot of people are wearing multiple hats. So our team's very small right now. We have 12 people doing all of this, analytical, QC, manufacturing, MSAT. So all of those teams are usually one or, one person does two jobs. So,
Right.
but as we're growing, as there's need, we're going to expand our team. And so part of the money that we receive from New York State, we are obligated to grow our workforce to 61. So as I mentioned, we're 12, so we still have quite a few hires over the next 5 years. So it's a good problem, right? And
Well,
I think
I like that framing of we're obligated to hire to 61. That's a great obligation to have, right?
it
Build
is,
out that
yes.
team. Yeah.
Yeah, and it's contingent. Our funding is contingent on the hires. So
Oh, that's great. Yeah.
yeah,
So it make like that's the first time like funding actually makes sense to me.
yeah,
Yeah, we want you to build something and we know you're going to have to hire to build. So that's part of the picture. So,
yeah. And it's really great that we had this track record in the beginning. So we were able to have these partnerships and we have talented faculty that are developing these trials. So we already have business set up. As soon as we open, we're going to have INDs approved and we're going to start manufacturing. So again, it's a good problem to have. And right now it's about execution. And that's where it helps having a lot of operations folks in addition to the scientific folks.
absolutely. And you're right. I think the proximity to the science folks, the more I think about what you said there, the more that resonates for me. Just because at this point, like maybe we're beginning to understand the rules of blood cancer CAR T,
Mhm.
like what the cells have to look like, what mix of I don't know CD8 cells should be in,
CD4, yeah.
in the final bag, like whatever it is. But I feel like we're just at the precipice for solid cancers of figuring out some of that. And so having that connectivity back to, your science expertise there allows you to sort of circle back and iterate and figure that stuff out. Is that how you guys are thinking about it?
Absolutely. That's exactly how we're thinking about it.
Yeah.
With solid tumors, I think we have a couple of cell therapies that were recently approved, both Iovance and Adaptimmune for solid tumors. So I think as we learn more about cell therapy, CAR T, NK, et cetera, and we learn more about solid tumors and how these therapies work in concert, or I shouldn't say in concert, or how we can fight solid tumors more effectively.
Yeah.
I think having both the operations folks and the science folks housed in the same area is really helpful. And it's funny at Kite, we understood this concept. So we have these facilities we call T cell factories, and then we have these facilities where you have research, the clinical team, the operations team all together in one site, and that's called RDMC. So research, development, manufacturing, and clinical. And so we had RDMC East and RDMC West. And so I worked on both. I ended up becoming the PD site head at RDMC East during my time there. So coming back to Roswell, where you have something similar,
Right.
it really resonated with me what we're building here at Roswell.
Absolutely. And I think it's funny because I try to describe this, particularly to friends in sort of small molecule world. Like, yeah, everyone has a role on these cross-functional teams that we all work on, right? Everyone has a role. And yet the way these products have to be developed, we have to be like intensely curious about everyone else's role and everyone else's business. We're not getting into each other's business. We're not like insinuating ourselves in, but we have to be intensely interested in the decisions that are being made by, research should be interested in CMC's decisions, CMC and clinical, et cetera, in part because these products are so complex
Mhm.
that you can miss a nuanced decision and its impact elsewhere if you're not being intensely interested in everyone else's function. Has that been your experience as well?
Absolutely. It's like you're reading my diary.
Like
Uh,
I don't want to know everything I know about qualifying an analytical assay, but I know a shocking amount. Right. And it's not because I feel like I need to make those decisions, but I know that like how far we push it or don't push it at certain stage of development can actually have ripple effects later down the line. And I bet you feel the same way about research decisions.
I think this goes back to having a robust operating model and
Mhm.
these decisions. Essentially, we're talking about governance, right? How decisions are made, and then how operations are done. Having these cross-functional teams, these matrix organizations, right, is very important because outside, so vein to vein, definitely we're looking to reduce that. Very important, but also the innovation cycle, right? How do you go from ideation to first in human? Because at the end of the day, we do these clinical trials and we don't know if they're going to work or not. That's why you do the clinical trials. So having, I think, a team that works together, having clear decision paths, really helps that innovation cycle going from ideation. So if you have an idea and it takes you 5 to 10 years before you get into human, I mean, whatever you can do to get, a lot of trials never get first in human. So
Right.
whatever you do, great kudos to you. But if you can make this streamline, and if you have an idea and get into first in human as soon as possible, let's say 2 years instead of 10 years, think of the benefit to the patients and, as a whole. So that's really our concept here is we really want to take that industry approach. So we have that operating model and I think you're on the program team. Is that
Yeah.
right? And then so that's strategy, right? Program team is strategy.
Okay.
And then you have operations teams, right?
Yep.
Or CMC teams, and that's really about
Yep.
execution. So really, you need that tie when you look at matrix organizations from the program team. Then you have the operations teams or CMC teams, and then even above that is okay. Where's the single source of truth?
Right.
And that's who they are, the single source of truth. Then it goes down to okay, then who's going to make the decisions? If you
Right.
can't make it within the functional teams or on the ground floor, then who's going to make that decision? So that's where you have the escalation path. So 100% agree with you. All of this structure is important. I think when you're looking at you want to help patients as soon as possible and you want to help get these therapies out there. Really reducing that innovation time is critical. And that's one of the reasons why, a lot of the faculty and folks came here to Roswell Park to help build this infrastructure, to help, scientists, companies, startup companies, as well as large pharma and biotech to come to Roswell, and we can help them really decrease that innovation cycle or innovation time.
That's fantastic. All right. So we're at the end of our time. So now I get to ask my favorite final question, which is if you could wave a wand and fix one thing and gene therapy development, what would it be?
Yeah, thank you so much for asking that question. And it really is the supply and supply chain and cold chain. So again, when you're looking at these type of products, they have to be shipped in dewars, or they have to be with allergenic products. Again, they have to be stored in liquid nitrogen. And even for the manufacturing process, sourcing the reagents and cytokines, et cetera, there's not a lot of companies that actually manufacture these in GMP for commercial. I think having people work on the supply chain aspect
Mhm.
of cell and gene therapy and having multiple sources of these reagents and critical components that I think would really advance cell and gene therapy in the future. And one of the things that I'd like to maybe touch upon is that old saying, right? An ounce of prevention is worth a pound of cure. And when you're looking at the future of cell and gene therapy and therapies in general, the ideal would be, hey, here's an injection or here's a pill. There you go, you're cured. So how do we get closer to that point? And I think supply chain is one of those aspects that'll help us get closer, and not just supply, even sourcing and procurement.
Absolutely. Yeah, no, I think they are oftentimes the unsung heroes of, I mean, how many times have we been clinically staged programs where we're like, what do you mean we're out of bags? What do you
Right.
mean? What do you mean the serum changed? What do you
Yeah.
mean? Yeah. So many times, right?
Yeah.
And even if you have, a deeply skilled person trying to navigate this, these products take I don't want to know what the list of key raws and other supplies are for a single manufacturing run. It's complicated. And so I love that idea. Having a more robust supply chain would absolutely be amazing. Thank you so much, Chris Choi, for joining us today. Really, really enjoyed this conversation. And best of luck to the Roswell team there. It's very exciting what you guys are getting to build, and I look forward to hearing about all sorts of success moving forward.
Yeah, thank you for having me. And please keep an eye out for our facility. We're looking to be up and running with our new facility as of March of 2025. And again, we want to be a one-stop solution. So for all the listeners out there, we're looking to partner, and our facility is ready to be used. So thank you so much for having me, Emily.
Thank you, Chris.